Welcome back. This is Cell Biology MIM Institute series, lecture number 3, and today we are discussing amino acid chemistry and proteins. So amino acids are the basic units of proteins, or the fundamental structures of proteins. Thus, proteins are heteropolymers of amino acids, and there are about 20 of them which are found in proteins. These are standard amino acids. However, some books, depending on their research and on their confirmations, they have said there are about 21, but most books are about 20. So proteins are high molecular weight compounds found in living cells. They contain carbon, hydrogen, oxygen, and nitrogen. Most contain sulfur. Some contain phosphorus, others contain iron, iodine, copper, manganese, or zinc. Some proteins serve as catalysts or enzymes, some as hormones, others as structural compounds with cells, and still others in physiologically significant functions such as ion transport, acid-base homeostasis, muscle contraction, and antibody-antigen reactions. So the 20 common amino acids shall soon be listed. The biologically significant amino acids are not found in proteins, as we pointed out earlier on. There are several other amino acids not generally associated with proteins, and there are above 300 amino acids in nature. So we shall first classify these amino acids. The first class is the nonpolar aliphatic aryl groups. The other one is aromatic aryl groups. Polar uncharged aryl groups. Positively charged, that is basic aryl groups. And the negatively, that is acidic charged aryl groups. So under the nonpolar aliphatic aryl groups, we have glycine, alanine, valine, proline, leucine, isoleucine. Remember, when you are asked questions about amino acids, the amino acids, it shall be very important for you to know the structure of glycine being the simplest of amino acids. You also need to know the 3-letter code and also the single letter code of these amino acids. Then under the aromatic aryl groups, we have phenylalanine, tyrosine, tryptophan. Under the polar uncharged aryl groups, we have serine, threonine, cysteine, methionine. Remember, cysteine is very important in the sulfur bonds, so you need to know this one. Then we also have asparagine, glutamine. Positively charged basic groups, we have lysine, arginine. We also have histidine. This one you also need to know it because it is a unique amino acid. Unique in the sense that it has a cyclic structure in it, while the rest have straight chain carbons. Then under the negatively charged acidic aryl groups, we have aspartic acid, glutamic acid, So you need to master all these groups. They are very, very important. Now, before passing over to other things, we also have non-standard amino acids which are found in certain types of proteins. For example, we have 4-hydroxyproline, we have 6N-methyl-lysine, gamma carboxyglutamate, desmosine, selenocysteine, etc. So let us go over to the properties of amino acids. The properties, we shall discuss them as subtopics. For example, under stereoisomerism, solubility, isoelectric point, titration of amino acids. So under stereoisomerism, the alpha carbon of each of the amino acids except glycine are each linked to 4 different chemical groups. This is characteristic of the asymmetric carbon, and that point just mentioned really describes why this particular amino acid, that is glycine, is really, really unique. So they are called alpha because the groups of interest H and NH2 are attached to the alpha carbon. Most of the naturally occurring alpha amino acids are of L configuration. For example, L-serine. The N, but there are situations where the D isomers also exist in nature, e.g. the antibiotic gramicidin and the cell walls of certain bacteria contain D amino acids. Some amino acids, e.g. histidine, exhibit dextro and levodopa depending on the pH of the environment. Properties: solubility. Amino acids exhibit a wide range of solubilities. Proline, for example, is soluble to the extent of 162 grams out of 100 grams of water at 25 degrees Celsius, whereas cysteine is soluble only to the extent of 0.011 grams out of 100 grams of water at 25 degrees. In general, amino acids are minimally soluble at their isoelectric point. So the property isoelectric point. In an electrophoretic system, amino acids migrate to the pole of opposite charge to that of the ionic state or species in solution. Since amino acids have both amino and carboxyl groups, they are ampholites and may be either positively or negatively charged. There is a characteristic pH at which the amino acid has no net charge. This pH is called the isoelectric point. At this pH, a monoamino monocarboxylic acid is completely ionized, but the charges neutralize each other. The dipolar ion formed is called a Zwitter ion. Titration of amino acids. The titration of either the carboxylic group or the ammonium group have been used as a means of quantitative determination of amino acid. Titration from pH of 6 to 12, however, presents certain technical difficulties which can be surmounted by resorting to formal titration. Neutralized formaldehyde solution added to a neutral solution results in a distinctively acidic mixture. Formaldehyde reacts only with the free amino acid group, not with the ammonium ion. The ionization of amino acids. Titration curves for proteins are divided into three pH regions. The pH that ranges from 1.5 to 6 corresponds to the ionization of carboxyl groups. The pH that ranges from 6 to 8.5 is attributed to histidine residues and terminal and alpha amino groups. Now, the pH ranging from 8.5 to up are included in the amino acid groups of lysine, the phenolic group of tyrosine, and sulfhydryl group of cysteine. Usually, the guanidinium groups of arginine have pK too high for observation in the titration curve. The pK values of ionizable groups in proteins may be modified by such factors as the ionization of other groups in the proteins, the proximity of hydrophobic residues, and the participation of H bonding in a physiological environment. Proteins are in the pH range of 6 to 8.5. Protein chemistry. We can classify proteins according to their biological roles. For example, enzymes, transport proteins, e.g. lipoproteins, nutrient and storage proteins, for example, seed proteins of wheat, corn, rice, or ovalbumin of egg, white, casein in milk, and ferritin in some bacteria, plants, and animal stores, iron. They can also be classified as contractile or motile proteins, for example, actin and myosin, structural proteins in tendons, cartilage, collagen, defense proteins, for example, immunoglobulins or antibodies, fibrinogen, and thrombin, regulatory proteins, e.g. hormones, insulin, G proteins, etc. So here is a short summary of classification based on biological roles. One, as enzymes, they have a function which is catalytic. As transport proteins, they include lipoproteins, nutrient and storage, contractile, structural, defense, and regulatory. Classification based on protein structure. Some proteins consist of a single polypeptide chain. Some are called multi-subunit proteins. They have two or more. If some individual polypeptide in a protein are identical, the protein is called oligomeric protein, and the subunits themselves are called protomers. Ribonuclease has one polypeptide. Hemoglobin has 4 subunits. 2 alpha are identical and 2 beta are identicals. Thus, hemoglobin is an oligomeric protein. Approximate number of amino acids residues in a protein, a simple protein containing no other chemical group. This can be calculated by dividing its molecular weight by 110, that is, give the molecular weight of the protein, the number of amino acids in it can be calculated. For example, bovine insulin, given a molecular weight of 5,733, number of amino acids in it can be found by dividing the 5,733 by 110, and it gives us 51 approximately. You can as well do for other amino acids. Here is an exercise. Calculate the number of amino acid residues in egg white lysozyme, which has a weight of 13,930. The human globulin, which has 64,500. The number 110 was arrived by taking the average molecular weight of 20 standard amino acids as about 138. The smaller amino acids are predominant in proteins, thus, when weighed for proportions in which the various amino acids appear in protein, the average molecular weight is nearer to 128 because the molecular weight of water is 18 and is removed for each peptide bond created. Then the average molecular weight of the amino acid residue of a protein is 110. So from here we are able to see that though biochemistry is truly a pure science, it's also not an exact science, because you do not really get something exact, as in this instance. The gaps can be seen, and that should leave room for error. But remember, biochemistry is a very, very, very good science, just as cell biology is. So classification of proteins, basing on composition. One, we have simple proteins. These contain no other groups except amino acids, e.g. the enzymes ribonuclease, chymotrypsinogen. Conjugated proteins, these contain amino acids plus some other chemical components. The non-amino acid group is called its prosthetic group, e.g. glycoprotein that has carbohydrates, amino acids as its prosthetic groups. And an example is immunoglobulin G. Now, the next thing we discuss is the protein structure itself. The amino acid sequence of a protein is inextricably linked to its function. So under the structure we have the primary structure. This simply relates to the amino acid sequence, can be determined in 4 ways. One, hydrolyze and determine amino acid composition, type, and amounts. Then you determine the N-terminal amino acid polypeptide protein. For example, you react it under the catalyst of 6 molar hydrochloric acid, and it gives you free amino acids. And under HPLC or ion exchange chromatography, you get amino acid composition. The other way is by use of Senger's reagent, that is 1-fluoro 2,4 dinitrobenzene. Now, sequencing of an entire polypeptide is done by Edman degradation procedure, that is from the N-terminal. Secondary structure of proteins, the alpha helix and beta conformation. Linus Pauling and Robert Corey were instrumental for the secondary structure of proteins. For this consideration, note that we have 2 types of proteins: the fibrous proteins, which contain largely a single type of secondary structure, and the globular proteins, which tend to be structurally complex. Most studies on secondary structures thus use fibrous proteins. Under this, we have the alpha helix. We also have the beta conformation, under which we have the beta pleated sheets. and also the beta trends. The tertiary structure is a three-dimensional arrangement of all atoms in a protein common with globular proteins. It concerns mainly folding, the formation of bends in the polypeptide chain during folding, and the direction and angle of these bends are determined by the number of location of specific bond producing amino acids as threol residues, etc. Then the other structure of a protein is the quaternary structure. This involves assembly of units. Now let us turn to a formal discussion of quaternary structure of proteins. Some proteins contain two or more polypeptide chains or subunits. For example, hemoglobin, RNA polymerase, aspartate, transcarbamylase, pyruvate dehydrogenase. The arrangement of proteins and protein subunits in three-dimensional complexes constitutes the quaternary structure. The interactions between the subunits are stabilized by multiple non-covalent interactions. We will not go into so much details about enzymes, as that is part of biochemistry and non-cell biology. So let us briefly discuss enzymes. Enzymes are catalysts responsible for driving chemical reactions within the cell. As catalysts, enzymes shorten the time required for equilibrium to occur in a reaction. They do not, however, change the equilibrium between reactants and products. They are proteins with the exception of some few catalytic RNA molecules, and in most cases are specific in their activity. This specificity is a property of the complex protein structure of the enzyme. The group of amino acid residues involved in the interaction of the enzyme with a substance is called the active site. It is the chemical nature, the number and configuration of these amino acids that determine the specificity and catalystability of the enzyme. If the active site is chemically modified, the specificity of enzyme may be lost, or the enzyme may cease to be a catalyst altogether. So some enzymes are made up of only amino acid residues. Some others need groups for activity. Thus we have a protein plus a cofactor. Here you have a holoenzyme. Cofactor is equal to inorganic ion, for example, an ion, magnesium ion, etc., or organic molecule called a coenzyme. So if a cofactor is covalently bound to a protein, it is called a prosthetic group. The protein part of the holoenzyme is referred to the apoenzyme or apoprotein. The primary, secondary, tertiary, and quaternary structures of proteins are essential to their catalytic activity. So our last portion on enzymes is classification of enzymes. So enzymes are classified according to the international code as given below. So number one, we have the class of oxidoreductases. An example is hydrogenase oxidase. The type of reaction catalyzes transfer of electrons. Number two, the class we have transferases, and an example is methyltransferase, transcatolase, amino acyl transferase, and the type of reaction is group transfer reactions. And number three, we have hydrolysase. An example is esterase, lipase, phosphatase, nucleases, cholinesterase. And type of reaction catalyzes hydrolysis reaction. And number 4, we have the class of lyases, an example of decarboxylases, carbonic anhydrase, aspartate, ammonia lyase, cysteine, disulfur and the type of reaction is addition of groups to double bonds or formation of double bonds by removal of groups. Number 5 is isomerases, and an example is racemases, epimerases, cis-trans isomerases, intramolecular oxidoreductases, transferases, and the type of catalytic reaction includes transfer of groups within molecules to yield isomeric forms. Number 6, which is the last but not the least, the class is ligases. An example is pyruvate carboxylase. The type of reaction is formation of C to C bond, C to S bond, C to O bond.